Good afternoon, everyone. Welcome to the Vanderbilt Microbiome Seminar Series. I'm glad you're all here. It's actually a perfect day to be inside as the rain hits Nashville. And we get to welcome a recent Sloan Fellow and Assistant Professor, Nick Shikuma from San Diego State University. It's safe to say that I and many others have been following Nick's work for years now. And in 2019, I'm not sure I've told this to him yet, but several of us were with him at a Gordon Research conference on Symbiosis, where we think he gave the most interesting podium seminar of the entire GRC conference, bar none. It was unprecedented, it was memorable, and I've been hoping we could get Nick to speak to our community for quite some time now, and so thank you for joining us today. Nick studies animal microbe symbioses across the scales of life with support from several private foundations, including the Sloan, as mentioned, and the Gordon and Betty Moore Foundation and federal funding. He carries some major funding levels, and his lab has discovered the first protein from bacteria that stimulates animal metamorphosis and tiny microbial syringe-like structures that inject toxins into animal cells, opening up the possibility for potentially novel drug delivery systems. So he was recently recognized as an invitee for the Early Career Scientist Perspective Special Series by M Systems, which is one of the ASM journals. And his free time, when he has it, is spent camping and going to the beach, one of the perks of San Diego, with his family. So welcome, Nick, and many thanks for joining us today.
Thank you so much, Seth, for that really flattering introduction. It's a pleasure to be here. Happy St. Patrick's Day to everybody. And please do interrupt me if you have questions along the way. I know it's kind of a strange format because we're online, but I'm happy to be interrupted, or I'm happy to take questions at the end. So please do just jump in if you have anything. Okay, so today I'm going to tell you about a tale of phage and tube worms. So this is our study of how we found marine bacteria stimulate tube worm development. And then we'll give you a taste of what we're doing right now in terms of what we found out with a pretty obscure model system and how that led us to study human gut bacteria. So starting in a broad context here, and on the x-axis, I'm showing in billions of years. And so this is the timeline of life on Earth as we know it. Well, what's thought is that the origin of life started about 3.45 billion years ago. And it wasn't for about 3 billion years later that animals came on the scene. And so between this time when life originated and then animals evolved, life had been diversifying, evolving, changing, but only in a microbial context, mostly. And so by the time animals got there, microbes had literally covered the Earth. And during this time of evolution in form and function that animals diversified, they did so in the context of microbes. And so perhaps the take-home message of this slide is just that we have never truly been alone in our evolution and development. Okay, and so now that we're here and we're really interested in studying how bacteria stimulate this host development, there's been a lot of new interesting models that have come online in terms of studying how microbes influence that development. For example, the Hawaiian bobtail squid, where microbes specifically colonize a light organ and then help that organ develop, allowing the bobtail squid to live out its lifestyle and ecology. And of course, most people probably know about tissue development in different mammalian species where the gut microbiome is helping the gut lining tissue develop. And so when you look at all of these different model systems, you can see that there are probably some overarching questions that we all study in terms of how these microbe-animal interactions take place. The first one is which microbes are important. And so that's probably a big question for everybody. There are many, many microbes out there, but it's probable that there are only a select few that are having the major impact on the host. Second, what are the mechanisms of communication? So once we identify those specific microbes that are important, how is it that the microbes are communicating with their host? Are they producing products or factors that are being perceived by the host and then being responded to? And then at the same time, how is the host coming back to the microbes and shaping the community and shaping the physiology of those microbes? And then once we study which microbes are important, identify mechanisms of communication within these specific model systems that we've studied, are there conserved themes that arise from studying many of these model systems? And can we identify ones that cross platforms or cross models that we can see occurring in these model systems? Okay, so now, of course, I'll zoom in on this particular one here, which is metamorphosis, which is what I study. So that involves these bottom-dwelling invertebrate animals. And so this can be, for example, barnacles or little biofouling tube worms that I study on the bottoms of ship hulls, or corals and coral reefs, or animals that colonize hydrothermal vents, like these giant hydrothermal vent tube worms. Now, these are the adult animals that we probably think of when we think of these types of animals, but they also have a biphasic lifestyle where they form free-swimming larvae. So these larvae swim around in the water column and allow the adult animals, which are stuck in place, to disperse to new habitats or replenish existing populations. And so these are microscopic animals that develop in the water column, and then at some point, they undergo metamorphosis. Just like a caterpillar undergoes metamorphosis into a butterfly, these larval animals undergo metamorphosis into this juvenile and then eventual adult form. And what's important to note about this metamorphosis is that it's irreversible. And so once this larva decides to undergo metamorphosis, it can't revert back to the larval form and swim away. And so making a decision of where and when to undergo metamorphosis on the seafloor is absolutely important for them to live out their adult life and then reproduce again. And in order to pick a good place to undergo metamorphosis, they use cues from their environment that tell them that this is a good place to undergo metamorphosis. And it turns out that bacteria have been one major cue that people have identified as a predictor of where these larvae are going to undergo metamorphosis. Okay, and so this phenomenon of bacteria-stimulated metamorphosis was first described in the 1930s by Claude Zobel and Esther Allen, right here in San Diego. But since that time, it's been almost 80 years, people have not really figured out too much in terms of what are the bacterial products that are stimulating metamorphosis, and likewise on the animal side, how is the animal perceiving these bacteria and responding to them by undergoing metamorphosis. Okay, so just to bring this point home here, the importance of this interaction could be that you can imagine that the bottom of the ocean is coated with bacteria, and these larval animals are using the bacteria as a cue to undergo metamorphosis on the seafloor. This could in part dictate animal biogeography. So you could imagine that on the seafloor, there's a heterogeneous distribution of bacteria in the environment, and that could in part dictate where animals undergo metamorphosis and then form potentially whole ecosystems like coral reefs. And it's also a widespread process. And so here I'm showing larval animals from sea urchin, a coral, and a tube worm. And so these are representatives of animal species that are from entirely different branches of the animal tree of life. Okay, and so just to show you that, here's a representation of the animal tree of life, where we've got cnidarians, corals, tube worms in the protostomes, and then urchins in the deuterostomes. And I've highlighted here in blue those clades in which there are representative species that have a larval form and they undergo metamorphosis into a juvenile form. So metamorphosis, as you can see, is widespread across the animal kingdom. Then in orange, I'm showing those clades in which there are representative species that undergo metamorphosis in response to bacteria. So again, you can see this phenomenon of bacteria-stimulated metamorphosis occurs in all different types of animals from very different clades of the animal tree of life. But again, what I told you is that this process of bacteria-stimulated metamorphosis is not very well studied. And one hypothesis I have for why this might be the case is because of the classic model historically studied in terms of metamorphosis. And these are Drosophila, Xenopus, the clawed frog. And these have been fantastic model organisms. People have won Nobel Prizes by studying their development and evolution. But the fact of the matter is that, as far as we know, these animals are not induced to undergo metamorphosis by bacteria. And so that means that we might have overlooked a very significant phenomenon of bacteria-stimulated metamorphosis because we've only studied a few model organisms. All right. Okay, so now we can overlay those grand challenges that I talked about in the beginning over this particular model system here. We know that there are specific microbes out there that are particularly important for stimulating metamorphosis, and we want to know which ones those are. We want to know the mechanisms of communication. What are the products that the bacteria are producing that stimulate metamorphosis? And then how are the larvae perceiving these products or signals? And then are there conserved themes that arise from studying this process? Okay, so first I'll tell you about a model symbiosis that we've been developing. This involves a tube worm called Hydroides elegans. The larvae, so this is a larval form here, and here's the juvenile form here. This larval form requires bacteria to undergo metamorphosis. And so what I mean by that is that if you keep their culture water clean and their glassware clean, they actually won't undergo metamorphosis into this juvenile form unless they see a substantial biofilm. And also we can produce hundreds or thousands of these guys in the lab at any given point. Metamorphosis is also a clear developmental readout for these particular worms. And so obviously this larval form looks very different from this juvenile form, and this entire process of metamorphosis takes place in under 12 hours. So here I'm going to show you a time-lapse movie of that metamorphosis taking place. So you can't see it, but this slide is covered with a thin film of bacteria that's stimulating this particular larva to undergo metamorphosis. This larva is losing the swimming cilia that's found around its neck, forming a calcium carbonate tube that it's going to live out the rest of its life in, and then also forming branchial rudiments on the top of its head. And so this whole process takes place under 12 hours. And so this forms the basis of one of our main assays that we perform in the lab, where we expose the larvae to specific strains or mutants of bacteria. And we might set that up in one afternoon. And then the next morning we come in, because this metamorphosis takes 12 hours to happen. We come in the next morning and we count how many of these animals have undergone metamorphosis from the larval form to the juvenile form. Pretty easy assay and really nice for undergrads actually to perform in the lab. Okay, and so on the flip side of this interaction are the bacteria. And there are many bacteria that induce metamorphosis, but not all of them do. And so, and they can be in many different types of bacteria. So gram-positive bacteria, gram-negative bacteria, many different types of gram-negative bacteria. And even bacteria within the same, categorized as the same species, you can have some strains that do induce metamorphosis and some strains that don't. And so that means that there are probably particular properties about specific bacteria that are important for stimulating metamorphosis. Okay, so the particular one I'm going to tell you about is called Pseudo-Ultramonas lutea violacea. This is the one that's producing violacean fleece. It's a strong inducer of tube-worm metamorphosis, but it's also been shown to induce the metamorphosis of corals and urchins.
Hey, Nick, we got one question. Go ahead, James. You can ask it if you're there.
Yeah, my question is more to understand the best topic. Like, do you say there are so many bacteria that induce metamorphosis? Do they induce the metamorphosis because each of these bacteria actually interact with the host in a reciprocal fashion that and co-evolve so as to induce the apoptosis, or is it just that they induce a stress in the host, like we are stressed by infections, and as such, any stress will induce the apoptosis?
Yeah, it's a great question. I'm going to say we don't know the answer to that, but it's really, we're just now getting into a little bit about what the tuberome response is, and it does look like it's certainly a signal transduction cascade that's post-translational. I'll tell you a little bit about that later in the talk, but it's possible that the bacteria are, it's not a kind of just general component of bacteria as far as we know. So for example, it's not like it's LPS, which most bacteria or gram-negative bacteria should have, that are stimulating metamorphosis. It seems like it's something very particular to specific bacteria, and exactly how, and as far as the story that I'm going to tell you, it's not a particular stress per se, but it's an actual toxin that's doing it. But we don't know in other cases whether it could be something different. But it's a really good question. And maybe we can follow up at the end of the talk once I've told you about the particular story that I'm going to tell you about, and then we can kind of talk about it more, because it's a really intriguing question in terms of like that different types of bacteria could have different mechanisms. So I'll tell you about that in a second, if that's okay.
Yeah, thank you.
Thanks, Nick. One quick question that I think you're going to get to later, which is, are bacterial metabolites or lipids enough to induce metamorphosis, or are the alive bacteria necessary?
Yeah, great question too. There are specific metabolites that you can isolate from bacteria, like and purify, that are able to stimulate metamorphosis. And that's a really good question, because it's possible that there are, well, it's a question because we don't know whether specific metabolites are, if you will, bioavailable from an intact bacterium to interact with the tuberome larva, versus just purifying them and having a big dose of a purified single molecule stimulating metamorphosis. So there's a lot of things that kind of stimulate them, but they're certainly not things that you would find in the environment. Like if you just add lots of calcium to the seawater, they'll undergo metamorphosis. But it's probably not the case that that's probably like a big stress that's just kind of like circumventing the normal pathway. And so it's not clear when you have a purified product if it's a circumventing the pathway, or if it's an actual like real stimulation.
Great, thanks.
Cool. Awesome. Yeah, please.
Oh, sorry, Dr. Shikuma. Following up to that question, what happens if you have like lower levels of concentrations of that purified toxin or whatever?
Yeah, it's a great question. So it seems as though, so we're working with non-isogenic populations of larvae. So these are wild-caught larvae, if you will. And so we see that there's a gradient usually in the stimulation of metamorphosis. So it's just, say if you have 100 larvae, if you back down the concentration, you'll just have fewer and fewer of them that'll undergo metamorphosis. And so it's possible that there's kind of the threshold for stimulation might be a little bit different in each of the different larvae, depending on the genetic background. But again, like that's a really good question, and we're not, we're pretty far away from answering it because of the how early we are in knowing a lot of this stuff. But really good question, and one that we've thought about a lot. Okay. All right, awesome. Thanks so much for the questions. Please keep them coming. Okay, and so let's see. We have P. lutea violacea, and it induces tuber metamorphosis, but different strains of this species have also been shown to induce the metamorphosis of coral larvae and urchin larvae. And so that means that it's a really nice model to study what are the bacterial factors or products that are stimulating metamorphosis. Okay, and the bacterium is genetically tractable, and it produces these things that we call metamorphosis-associated contractile structures, which are the things that are stimulating tuber metamorphosis. So this is a story that started out as when I was a postdoc in Diane Newman's lab, along with a collaboration in Grant Jensen's lab and Mike Hadfield's lab. Mike Hadfield was the one that initially started this tubermin bacterium system. Martin Pillhofer was a postdoc at the time, and Gregor Weiss was an undergrad. Okay, and so the prevailing model of how people think bacteria stimulate metamorphosis goes like this, where you have a bacterium that's formed a biofilm on the seafloor, and they're continually, through normal growth and metabolism, producing factors or products that might be soluble in the seawater or bound to the cell surface or perhaps bound to the biofilm surface. And animal would happen along sniffing out for these factors, which would then bind a cell surface receptor, initiating a signal transduction cascade, which would lead to metamorphosis. And it turns out that MACs are quite different than this expected model of how people thought bacteria stimulated metamorphosis. Okay, so it turns out that MACs are related to the contractile tails of bacteriophage. And this is a whole family of different structures called contractile injection systems. And they're all related to this contractile tail. So these are phage, which typically have a DNA-filled capsid, and then a contractile tail, which has a rigid inner tube, which allows puncturing of a cell membrane and then injection of the DNA into the bacterial cell. Turns out that bacteria have stolen the machinery in some cases from the phage, and they use it for their own purposes by having the tail anchored to the cell membrane and then shoot out that rigid inner tube into target cells from within the bacterial cell. These are called type 6 secretion systems. And then there's a whole nother type of these things called extracellular contractile injection systems, which are more like phage in that they have the contractile tail portion, and the bacterial cell fills up with the phage tails, just like it would with the replicating phage. The bacterium lyses, releasing the phage tails, and these go on to autonomously bind target cells and then inject their contents into target cells that way. So one example of extracellular contractile injection systems are MACs. And a little foreshadowing here. So contractile injection systems often inject protein payloads into target cells, which is how the extracellular contractile injection system or the type 6 secretion system performs its molecular function on that target cell. Okay, and so at the time we were really curious to see which type of contractile injection system we had, and we knew that the genes were present in the genome, and we knew that the genes were important for stimulating metamorphosis, but we didn't know what they were doing. And so we initially tagged the base plate here with superfolder GFP, and then did a nice time lapse to see how are these things produced within the bacterial cell. And so you can see that that cell that's pointed out with an arrow there, it'll come back around again, is going to become amorphous. So this is going to double here, become amorphous and lyse, releasing those phage tails from within the bacterial cell, and then you're going to see GFP forming in the place of where that cell lysed. And you'll see these other cells lysing and forming GFP in place of the lysis. Okay, and so here's a still image of what that looks like by fluorescence microscopy. And at the time, it was really curious because if you were to image a single bacterial or a single phage, it would be a tiny foci right next to a bacterial cell. But instead, what we saw were these 1 micron across donuts by fluorescence microscopy outside of the cells. We could quantify out of a population of cells how many of them were producing MACs within their cell and then lysing. And it turned out that about 2.4% of the cells lysed and produced MACs, at least by when you grow these cells in laboratory culture. And the rest of them would just stay intact. Okay, and so then at the time, the big question was, what do MACs look like? And to answer that question, that's where Grant Jensen's lab came in, where they specialize in a technique called electron cryotomography. What I'm going to show you is an electron cryotomogram, where we're going to pan through this image in a near native state at 4 nanometer resolution. And then we're going to reconstruct what we think these MAC structures look like in 3 dimensions. Okay, and so a nice story that goes along with this particular cryotomogram is that Gregor, the undergrad, was working really hard on the cryo-electron microscope late at night, because that's when you allow undergrads to work on these high-powered microscopes. working really hard, and you have to render these images on your computer. And so he acquired the images, threw them on his computer, was rendering them, and we had a meeting set up at 9 a.m. the next morning with the PIs and the postdocs and Gregor. And so he threw them on his laptop, ran over to the meeting without looking at the images, and then turned out that we were all sitting around the table. Gregor opened up his laptop, and we looked at this image all at once, sitting all together, and had this really wonderful holy crap moment when we first saw this pretty wonderful structure together. It's one of these really nice moments in a scientist's life when you have that like aha moment. So what are these things? These, we lovingly call them Death Stars in the lab, but this is because this is what they look like. This is about 100 phage tails all held together by the tail fibers, which are in orange, and this pretty odd hexagonal lattice. And so here's a slice of the cryotomogram where you can see that hexagonal lattice in a still image. The blue portions are the contractile sheath, and the green portion is the inner rigid inner tube.
Just a very quick question. Do CIS come from a prophage locked as a relic within a lightogen or not?
Yeah, it's a great question. We do think that they're a relic of a previous phage, and they've just been potentially co-opted. That's the theory, is that they've been co-opted from a phage, perhaps lost the capsid protein, and just are instead used as molecular weapons by the bacterium to perform their function.
It's really thrilling. Thanks.
Thank you. Yeah. So now we'll take a slice through this cryotomogram. So this is where you're going to see that it's pretty similar to a phage by cryotomography. So these are the both extended forms and contracted forms of the phage tails. So you could imagine that each of these is an individual phage tail. The sheath is in an extended form here, so ready to fire, or in a contracted form here with the base plate and the tail fibers and the rigid inner tube sticking out here, or fully shot out in this case here. And Martin and Gregor were able to perform subtomogram averaging and get really nice high resolution structures of the tail tip in both the extended form. So you can see the tail fibers here and the tail pin sticking off and the tail tip. And then also in the contracted form, you can see the 12-fold symmetry of the sheath and then the tail fibers emanating from that base plate. Okay, and so just to convince you that it really is these MAC structures that are stimulating metamorphosis, we could create cell-free extracts of the MAC structures from the cells. If you take that extract from wild type, you can get a lot of metamorphosis. So this is just our normal assay where we take larvae and we expose them to different strains of the bacteria. And after 24 hours, we count how many are in this juvenile form versus how many are in this larval form. If we knock out that same protein that we tagged with GFP, we can see that extract from that strain does not induce metamorphosis. And what I showed you is that these things are about 1 micron across by fluorescence microscopy. And so if we filter that extract through a 0.45 micron filter, we abolish almost all of the metamorphic properties of that extract by just a really crude filtration. So it really is, we think, these 1 micron across donuts that are stimulating metamorphosis. Okay, and so just a nice size comparison here. Here's a MAC structure by fluorescence microscopy that matches up really nicely with what we see by electron cryotomography. And if you were to put 1 micron next to a tubeworm larva, it's that little red dot underneath the arrow here. And so our extremely hand-wavy hypothesis at this point of why the MACs are making multi-tailed arrays of phage tails instead of a single tail is that instead of interacting with a bacterial cell, which is about 2 microns long, it's interacting with a giant tubeworm larva, which is 200 microns long. And so potentially it's multiplying its numbers by 2 orders of magnitude in order to interact with an animal that's 2 orders of magnitude larger. Okay, so now I've told you about our model and that we've identified a pretty intriguing secretion system that stimulates metamorphosis. And remember that I told you a little foreshadowing that contractile injection systems often inject protein payloads into target cells. And of course, once we found MACs, the next question was how do MACs induce metamorphosis? What are they doing to interact with that target cell? So this is a nice work by my master's student, Chip Erickson, along with Fabian Eisenstein, who's a PhD student in Martin Pillhofer's lab. So he was the postdoc that I collaborated with originally, and we continue to collaborate. He started his own lab at ETH Zurich. And then this involves Bob Zeller, who's a PI in my department, and I'll tell you a little bit about his role in a second. Okay, so we had Fabian perform subtomogram averaging instead of on the tail tip on the sheath and tube portion of this of the MAC structures. And when they performed modeling on this subtomogram average, they could dock in different proteins from the gene cluster into this predicted model. So you could see the blue here is the sheath protein that could be docked into the structure. The tube protein that's rigid, that's should be injected into the target cell. And there was also this curious electron density in the middle that we hypothesized was a cargo or a payload that the MACs were injecting. So you can see that electron density here. And when we were looking through the gene cluster, we actually found that a specific protein was particularly interesting, and we knocked that one out, or knocked that gene out, and found that indeed, if we knocked that gene out, we could see a loss of stimulation of metamorphosis. And when they performed subtomogram averaging and cryotomography, they found that the MACs form intact MAC structures, except the inner tube appeared to be empty. And when they performed subtomogram averaging, they could see again, the inner tube was lacked in electron density in the middle when compared to that wild type that had the electron density inside. Okay, and so, and just to bring it home here, MIF1, if we knock out this protein, it does not induce metamorphosis, even though I'm not going to show you the data. So then the question was, is MIF1 sufficient? Is that the thing that's loading the inner tube and actually getting injected to stimulate metamorphosis? Okay, so what Chip did was he created recombinant MIF1, and here's a Western blot showing we can create recombinant MIF1 inside E. coli. And this is where Bob Zeller comes in. So Bob Zeller has pioneered the technique of electroporation into Ascidian embryos. And so that whole field is taken off because he uses his electroporator to modify Ascidians. But we borrowed his electroporator that he calls Zeller zappers. And he actually makes these literally in his garage. So here's his custom electroporator that we borrowed, and we took our recombinant MIF1 and we tried to zap them into the tubeworm larvae. And so here you can see we've got recombinant MIF1 and a couple other of recombinant proteins that we've created in the same manner as the MIF1. And we can see that MIF1 stimulates metamorphosis when it's zapped in the tubeworm larvae, whereas the other ones don't. And so interestingly, if we just add MIF1 to the seawater and don't electroporate, we don't see induction of metamorphosis. We only see it if it's zapped into the tubeworm larvae. And so this follows with our model suggesting that maybe the MACs are actually injecting the protein into the tubeworm larvae, and then that's what's stimulating metamorphosis. Okay, all right, so this brings us kind of back to our model of what we think is going on. Here's P. luteoviolacea forming a biofilm on the seafloor. We think that a small percentage of those cells are turning on the genes for MACs. MACs are being produced within the bacterial cell, and the chaperone, I didn't tell you about that chaperone protein, but we think that chaperone is loading MIF1 into the inner tube of the MAC structure. MACs are, the cell is lysing, releasing the phage tails, which are embedded within the biofilm. And the tubeworm larva comes along, it's sniffing out for different factors from bacteria. It's injected by MACs. MIF1 is injected into the tubeworm larva, and then that signal transduction cascade occurs, initiating metamorphosis. Now, as you can see, this is a totally different mechanism from, or model of how we think metamorphosis is taking place in response to bacteria when we compare it to the original model, where people thought that there were factors that were, or products that bacteria were producing by normal growth and metabolism, and that was somehow sniffed out by the animals and then stimulating metamorphosis that way. But the nice thing about this new model is that actually we think they're not mutually exclusive. And so both types of stimulation can probably take place. We probably think that that actually is what's happening in the environment, where there's a more potentially animal-driven process where the animal is in charge, sniffing out specific metabolites from bacteria and then initiating metamorphosis on its own. But then you can have a more bacteria-driven process where the bacterium is producing a macromolecular machine, which is injecting a specific protein into the target cell and then initiating metamorphosis that way. So quite different than how we thought this process took place. All right. Okay, so
hey, Nick.
Yeah.
Do you have a sense yet of how specific these MACs have to be to this variant versus divergent variants in order to induce the metamorphosis?
By variant, do you mean like different types of animals, for example, or?
The structure itself, I'm kind of thinking about, well, do related bacteria have slightly divergent MACs?
I see.
In amino acids, proteins, and ultimately structure. And as you go further away from this type MAC, would you lose sensitivity to induce metamorphosis or not? It's more of a specificity question about, does it require this MAC versus other related MAC?
Yeah, great question. It turns out that MIF1 is very strange. And what I mean by that is that if you blast it, for example, in just the non-redundant database, you don't find very many homologs, which is pretty surprising. The E-value drops off pretty quickly after you get outside of the Luteoviolacea species. And we don't know if that's due to the fact that MIF1 is just evolving really quickly. And so maybe we lose signal because it's kind of like a phage where the phage proteins are evolving really quickly and we can't see it just because of the bioinformatics are not able to match up.
Right.
Or whether MIF1 is truly unique. I guess that's the other possibility, and we only find it in Luteoviolaceas. But that's a really great question. It's one that we're actually pursuing right now, where we are trying to find the exact point at which we're trying different species of pseudoaltermonads. And it turns out that many different pseudoaltermonads have the main machinery. It could be that the main machinery is more easily recognized bioinformatically. And so we can see lots of bacteria with the main machinery, but not MIF1. And so we're trying to figure out, is there a point at which they've lost that MIF1 protein, or where is it that they gained MIF1 essentially, and then gain the ability to stimulate metamorphosis?
Exactly. Thanks.
Yeah, super good question. One we're totally following up on that right now. Okay, so now we've identified this first protein from bacteria that stimulates animal metamorphosis. And now I'm going to move on. I'll tell you a couple stories about conserved themes that we've come across based on this model system that we're studying. Okay, so this is a work by Kyle Malter, my PhD student, an undergrad, Mimi Esmeralda, who's now a PhD student at UC Santa Barbara, and in collaboration with Erica Forsberg's lab and Mayadith Dampa here, who's a PhD student in her lab. Okay, and so the question then becomes, how does MIF1 induce metamorphosis in those two-worm larvae? And what I told you, or what I was kind of alluding to, is that MIF1 is highly evolvable, and it also has no identifiable domain. So this was like our first thing that we did, was just blast it, see if, or look for any domains. We tried every program we could think of, and we couldn't find any homology to other proteins, which is pretty, so that makes it difficult for us to really understand how MIF1 is functioning. but we did look in the literature, and we were intrigued to see that it turns out that metamorphosis in these marine animals occurs in a little bit of a different, or a pretty different way from normal metamorphosis that you would expect in, say, Drosophila and Xenopus. And the difference is that in Drosophila and Xenopus, it's a very transcription-based process where it's a prolonged metamorphosis, where metamorphosis takes place over days or weeks in order to happen. Whereas in this case, as I showed you with the tube worm, it happens within 12 hours. And so it turns out that for many of these larvae, undergoing metamorphosis is a pretty vulnerable part of their lifestyle. So they need to undergo metamorphosis quickly in order to survive. And so that means that they're probably using post-translational signaling mechanisms in order to initiate metamorphosis and then allow it to happen quickly in order to get into that juvenile form, form a calcium carbonate tube where they're going to be safe on the seafloor. Okay, and so that meant that we were looking for lipid signaling. So that's been shown in many of these different animals to be important for metamorphosis happening. And we also were looking at other conserved signal transduction processes that were post-translational, like protein kinase C and MAP kinase signaling. Okay, and so that meant that there's kind of this supposed signal transduction cascade that happens, and then ensuing developmental change that happens between the larval form and the juvenile form once MACs stimulate this process. So that's where Erica's lab came in. They specialize in LC mass spec. And so we took the wild type and the MIF1 knockout bacteria, and we exposed those larvae. And then because we wanted to see that window of time when they were initiating metamorphosis, we took a 1 hour time point after we exposed them to the different types of the bacterial structures. And here I'm showing a cloud plot of that LCMS run and a comparison between the wild type and the MIF1 knockout. And so here you're seeing retention time of on the LC mass spec, and then the mass to charge ratio of the different masses that were found. And we performed a lipid extraction. So we're specifically looking for lipid signaling components that might be important for stimulation of metamorphosis after this interaction with the MAC structures. And what was intriguing, so we found 33 significant features, but the most significantly differentially regulated lipid was diacylglycerol, shown here. And so that was intriguing because in the literature we were digging back, and diacylglycerol was also important in cnidarians to initiate metamorphosis. So we took that piece of evidence and we ordered diacylglycerol from Sigma, and found that if we just added diacylglycerol, DAG, even in the absence of MACs, we could get metamorphosis to happen. So here's metamorphosis on the x-axis or the y-axis, and then concentration of DAG on the x-axis. And so you can see that DAG is a really strong inducer of metamorphosis, even in the absence of any bacteria. And if you know this, this signaling cascade, diacylglycerol directly binds to protein kinase C to activate a signal transduction cascade. And so indeed, when we used protein kinase C inhibitors, we used a couple different types, although I'm just showing you one example here. Again, metamorphosis on the y-axis and concentration of the inhibitor on the x-axis. We could inhibit metamorphosis by, even though we added DAG, we could add the inhibitor at the right time and then stop metamorphosis from happening, because we think the signal transduction cascade is first going through DAG and then protein kinase C. And again, I'm not showing the data, but we have MAP kinase inhibitors that also work on the tube worms, and shown that it's this linear signal transduction cascade that's being initiated by the MACs to stimulate metamorphosis into this juvenile form. Okay, so MIF1 activates, here's the punchline is that MIF1 activates a linear signal transduction cascade that's initiating that metamorphic process. And if you know, PKC and MAP kinase and diacylglycerol signaling are all very well conserved signal transduction systems in many different eukaryotic organisms. So pretty intriguing that we could identify these signal transduction systems that are important for stimulation of metamorphosis in this tube worm. Okay, so now we've identified a secretion system, a protein that stimulates this metamorphosis, and a developmental program that initiates this process once MIF1 is injected into the tubeworm larvae. Okay, so now my final story here is a continued conserved themes. This is work by Marissa Rojas and Gisele Cavalcanti in my lab, and also in Forrest Rower's lab that's downstairs from me here. So what they did was they were interested in the diversity of these phage tails in different metagenomes. And so what they did was they took the sheath protein, which is this contractile portion here of the tail, and they recreated a phylogeny of different phages and contractile injection systems, which include type 6 secretion systems and also those extracellular contractile injection systems. So on this phylogeny, we can find phage T4. So this is the workhorse T4 that people have studied for a long time in terms of molecular biology. We can also find type 6 secretion systems that are known and initially identified. So these are from Vibrio cholera and Pseudomonas aeruginosa. A little bit more recently, people have found Bacteroides from the human gut have another type of type 6 secretion systems. And so if you're familiar with this, these are the subtype 1 and subtype 2 type 6 secretion systems, and these are the subtype 3 type 6 secretion systems. So they're a little bit more distantly related to these initially identified type 6 secretion systems, but these are intriguing because they promote bacteria antagonism within the human gut. So you can you see clearing of whole swaths of strains because they have certain effectors within their arsenal. Pretty intriguing. But what Marissa and Gisele found, which was surprising, was that MACs occur on this entirely separate branch of the phylogeny here. So, and what's even more surprising is that when we looked at the literature to look for other contractile injection systems that were related to MACs that interact with tube worms, we found that they interacted with insects, for example, grass grubs, and then also amoeba. So these are forming phage tail-like structures just like the MACs do, except at least right now, with the handful of studies that are in the literature, it seems as though all of these structures are mediating bacteria-eukaryote interactions instead of bacteria-bacteria interactions. What was also curious is they found that Bacteroides species had a gene cluster that looked a lot like the MACs gene cluster in P. lutea violacea. Initially, we thought that they were just these type 6 secretion systems, the subtype 3s, but it turned out that they fell on this side of the phylogeny, and they were much more closely related to the tubeworm MACs than to these subtype 3 type 6 secretion systems. And so we named these eventually Bacteroides injection systems because we think that they're a entirely new flavor of these contractile injection systems. Okay, and so if you look at the gene cluster from P. lutea violacea, this is the one that interacts with the tubeworm, and you look at the gene cluster, we can see different components within the gene cluster that encode for different structural parts of the structure. So we can find the tail tip, the rigid inner tube, the contractile sheath, and the base plate, tail fibers, tail pins. And then we mined 749 Bacteroides genomes to see if they had similar gene clusters. And what we found was that about 9% of that 749 had BIS gene clusters. So, and they came in about 3 different arrangements. So these are from Bacteroides cellulosilyticus as an example, or fragilis, or Parabacteroides distinatus. Okay, and so super intriguing that we could find these in isolates of Bacteroides and Parabacteroides, but then the question was, are they actually inside human microbiomes? And so we took a representative with all of the major components that compose these phage tails, and we looked in 2 different data sets that were available to us publicly. One was the Human Microbiome Project, which just took the microbiome of 215 individuals, and then this meta-hit project, which took the microbiome of 124 individuals. And these were done a fair time ago, and so all they were doing was just looking at the gene catalog of a normal healthy human microbiome and seeing what's there, and totally independently perform studies from people in Europe and also the United States. So when we mine those data sets, so here's Human Microbiome Project and MetaHit data set, and then the % individuals that have this many genes from this BIS gene cluster. And what you can see is that nearly all of the individuals at least have one of the BIS gene clusters, and it's probable that they have all of them. It's just that the sequencing depth was not deep enough to identify all of them in all of these individuals. And so that means that nearly all adults potentially possess BIS genes within their gut microbiomes. And perhaps you and I sitting here have MACs floating around in our guts right now, and we just don't know what they're doing. Okay, and so the last thing we did was mine another data set. So the Human Microbiome Project came out with a second version where they compared healthy individuals from 214 of those with people, the microbiomes with people with different diseases. And so this particular one was from people with inflammatory bowel disease, 103 individuals. And when we looked for those genes within those two populations, what we could find is that here I'm showing frequency of those within those two different populations and then number of BIS genes on the x-axis. And what you can see in gray is that there's healthy individuals have a lot of BIS genes within their gut microbiomes, whereas IBD individuals had fewer BIS genes within their microbiomes. That was super intriguing, but also we were a little skeptical and we thought, okay, well, IBD individuals probably have fewer Bacteroides and Parabacteroides in their gut microbiomes because those people often have dysbiotic microbiomes. And so we performed that, we reasoned that, and so we performed that comparison there. So here I'm showing on the x-axis Bacteroides and Parabacteroides relative abundance and the frequency of those individuals within those two different data sets. And intriguingly, we couldn't find a large correlation between differences between the two populations and the number of Bacteroides or Parabacteroides in their gut microbiomes. And so this correlation of healthy individuals having more BIS genes is not explained solely by the fact that they have different counts of Bacteroides in their gut microbiomes. All right. Okay, so finally, and my last conserved themes is that we've found a new contractile injection system gene cluster within human gut microbiomes. Okay, and so for the last couple of a minute or two, I'll just tell you about my team, acknowledging them as fantastic people in my lab. I couldn't have done any of this without their help, and also tell you a little bit about what we're up to currently. So we've got a fantastic group of students working on the MAC structures. There's a lot to do. These are novel structures. The multi-tailed form is something that we haven't seen in any other structures before. So we're certainly following up on that in collaboration with Martin's lab. Chip was the guy who has performed one of those. He performed the MIF1 study, but he's gone on to start his PhD in Martin's lab. So we've got a man on the inside doing our dirty work over there. We've got continuing to do work on MIF1 as I talked with in response to Seth's question. And then of course, we're looking at the host response. And so we found a couple of different intriguing pathways, but of course, there's way more to flesh out there in terms of how is MIF1 stimulating metamorphosis. We still want to kind of go in between. Diacylglycerol is probably just a secondary messenger in terms of initiating the signaling cascade, but we want to go upstream of that. Where is it that MIF1 is really initiating the cascade and stimulating metamorphosis? Another set of people that are looking at BIS. We've got Bacteroides in the lab now, and we're performing genetics to really look at what those structures look like and potentially what they could be doing in the gut microbiome. And then things that I haven't told you about, we've come out with a couple studies or ones that are coming out soon, which are about very different factors. And so even though MACs are these really intriguing systems, there are very different things that look like small molecules, for example, tetrabromopyrrole, that are able to stimulate metamorphosis still in the same tube worm, but an entirely different structure. So entirely different like class of molecules even, right? Small molecule versus a protein that's able to stimulate metamorphosis. Even though it's the same developmental pathway, totally different. And so, as I said, we're kind of leaning into this fact that there's probably a big spaghetti of interactions out there in the environment that are happening where multiple bacteria are making very different products that are stimulating metamorphosis, and they might look completely different even though they're stimulating the same process. All right, so that concludes my talk. Thanks so much for listening. And of course, I'm happy to take questions. Please do. I'd be happy to have a discussion for as long as you can. So.
Awesome. Thank you, Nick. Yeah, we've got a couple questions in the waiting. That was a tour de force in the life sciences. Very much appreciated. If we could all give him a round of applause virtually or actually turning mics on. Thank you. Okay, so I'd like to ask Anna to ask her question. Anna, is your mic on?
Yes, sir. Thank you so much for this talk. This was really, really interesting. I was wondering if there were any studies looking at like the evolutionary background of these mechanisms, like how long have these mechanisms been in place?
Yeah, good question. So are you referring to like the MAC structures specifically, or the
Yes.
Or metamorphosis?
The MAC structures. I was just curious if these kind of evolved recently, and if there were different mechanisms for inducing metamorphosis before that. It's kind of a broad question. I'm sorry.
No, it's perfectly. It's a really important question, and something that, of course, I guess we're not quite sure about. I can speculate. When you look for MAC gene clusters within lots of microbiomes, so we didn't just look at the human microbiome. We looked at lots of different environments, and it turns out that MAC gene clusters are everywhere. Like, so they're in terrestrial environments. As I said, they interact with grass grubs and amoeba, so they're interacting in a pathogenic context. You can also find them in a lot of different, very different types of bacteria. So for example, cyanobacteria have similar gene clusters. They've even been found inside archaea. And so you can actually at least see the gene clusters in archaea. Nobody's ever seen the structures, if archaea even make them, but super surprising that they're found in so many different types of microbes. What they could be doing, we don't know, but I guess that the fact that they're found everywhere means that they are probably quite ancient in terms of their when they originated, because they've been able to be spread around so much. One interesting tidbit about these structures is that it looks as though the structural components of these MAC structures have been passed around, but different microbes have swapped out the effectors. So in a closely related bacterium, you might have the structural components, but then have an entirely different effector that's loaded into that structure. And it looks as though the microbes are essentially swapping out effectors depending on what they need the MAC structures to do for them in their context.
Very interesting. Thank you.
Yeah, great question.
Right, let's keep. Yeah, I was just gonna maybe ask the next person, Dalton, to go ahead and ask a question.
Perfect. I guess I jumped the gun a little bit. I was excited.
You got it.
I've been the question for a little while. So, when you started talking about metamorphosis, I immediately started thinking about the strategies that they use to perform tissue engineering. So like printing human organoids, and typically they do that with putting some sort of stem cells in the right place and allowing for self-assembly, kind of follow like a morphogenesis pathway. But considering these bacteria-eukaryote interactions, how likely is it that bacteria will provide like some major beneficial aid in the self-assembly processing of these engineered human tissues?
Yeah, great question. What I suspect, although again, there's just not much known about this system, is that what we suspect, because there's a post-translational signal transduction that happens that's probably orchestrating most of the metamorphosis happening in these marine organisms, that they might be kind of primed in order to undergo metamorphosis, and perhaps all of the machinery that's required for the tissue development and like tissue rearrangement is present within the larval form. And then it just needs kind of a initiation of the signal transduction cascade, which is kind of probably permeates the whole animal. And then that starts off, say, a domino effect, essentially, which then causes all of that metamorphosis to happen in such a short time.
Mhm.
At least that's how we're thinking about it right now. But of course, that's all pretty speculative, because metamorphosis in marine animals is not very well studied at all. But yeah, great question, Dalton.
Okay, thanks so much. And thanks for the great talk.
Yeah, of course.
And Nick Markham has a question that I was also going to ask as well. It often comes up when I hear about this. Go ahead, Nick.
Thanks. Yeah, I was just curious if it's understood how the transformation and induction of metamorphosis is beneficial to the bacteria or bioform formation.
Yeah, it's a great question. And as Seth alluded to, it's one that comes up often. What people think is either that there's two possibilities. One is that they don't get any benefit at all, and the tube worm is just honing in on a signal from the bacteria. The other option is that perhaps the bacteria are, so there's kind of two possibilities in this possibility, where the bacteria could be stimulating metamorphosis in particular to colonize the host. So that has yet to be determined, whether they benefit from a tube worm that's metamorphosed next to them, say on the bottom of a ship. And that's probably pretty plausible, because these tube worms form really thick coats of tubes on the bottoms of a ship. And I would say that that's probably a really nice place for a bacterium to live. The other possibility is that they're creating these structures as a defensive mechanism. So lots of these biofilms are probably grazed by protozoans and things like that. And so it's possible that the protozoan, they're defending themselves, and then the tube worms have just identified these things as good cues in order to undergo metamorphosis in response to. And perhaps they're getting some benefit, because now that these pretty toxic structures that might be present around them, which could deter other things from undergoing metamorphosis around them, because these ecosystems are very competitive, because everybody wants to be on the surface, and they're forming these big crusts of different organisms. And so that's certainly possible as well. We're starting to dip a toe into that question as well, of looking to see colonization of the tube worm perhaps, or some kind of interaction with the tube worm that's past metamorphosis. But yeah, something really intriguing and something we're thinking about a lot. Great question.
And Sadiq, are you there to ask your question?
Yes, I'm here. Sorry.
Great.
So I was wondering, like, what could be the possible function of MAC in human microbiome? Like, is it any studies has been done, or is it completely unknown yet?
That's completely unknown at this point. We're totally exactly looking into that possibility. We have at this point a couple of guesses as to what the effectors are that are loaded into the structure, kind of going after those, and then also going after what the structure looks like. And one thing I didn't mention, which is intriguing, is that MACs, the ones from P. lutea, the marine bacterium, have tail pins that stick out from the business end of the tail. And so those, we think, are the triggers for contraction and injection of the tube. And it turns out that those appear to be specific for eukaryotic cell surfaces. And so it's possible that the MACs are specifically targeting eukaryotes because of, just like, phage have tail fibers that specifically bind their target bacterium. MACs might have a similar way of binding their host, but instead it's targeting eukaryotic cell surfaces, which is quite intriguing. And so we're also looking at that component of the BIS gene clusters because it's such an important part of the interaction. And so that might give us clues as to what they're interacting with. It's entirely possible that those BIS genes or the BIS are interacting with bacteria and not the host. And so we certainly can't rule that out.
Mm-hmm. Do you also think like it could be like associated with like disease and this kind of things? Like,
yeah, entirely possible as well. I think that the possibilities are totally open at this point. It'll be intriguing to see what comes out of it.
Thank you so much.
Yeah, thank you.
Okay, and Jennifer. Jennifer has a question. I'll go ahead and ask it in case she had somewhere else to go. Are MACs implicated in metamorphosis in any other species? Do they possibly play a role in cell differentiation in host eukaryotic gut cells?
Yeah, it's a great question. It turns out that MACs are kind of strange in the fact that, so we've tried them on a bunch of different types of organisms, and for some of them, they're very toxic. Which maybe is not surprising given the possibility that they could be defensive structures. But we put them on different cell lines actually in the lab. So we have insect cell lines and mouse macrophages, and it turns out that they're very toxic to those cell lines. But we also found that they have a effector, a second effector, so this is separate from MIF1, that's mediating that killing effect on the cell lines. And so they're able to interact with lots of different types of eukaryotes and have different outcomes for the different eukaryotes based on the effector that they have. And so potentially, we don't know what they're doing in other systems. So we haven't found MACs to be inducing metamorphosis in any other organisms, although we've only tried them in a couple of cases, I would say. So cnidarian larvae, we're going to try them on corals pretty soon. And potentially we also have urchins in the list, but not quite there yet. Yeah, so just kind of, that's definitely a super fun project for an undergrad to just throw things together and see what happens.
Absolutely. Okay, looks like James, we can have you as the last question.
Yeah, thanks. My question was more of ecological perspective. So with these experiments, you have proved that these bacteria using MACs can induce the change from the larval state to the adult state. But have you actually tried to see whether it plays a key role in the environment for the host species, or whether it's just an annex role in the environmental factors? Does the species rely on bacteria to survive through changing from the larval stage to the adult stage?
Yeah, it's a great question. We've kind of gone down that road in terms of looking at the composition of different bacteria in normal biofilms that you would find, say, on the bottom of a ship, to get a sense of what are the concentrations of different, or what's the composition of different bacteria, types of bacteria on in the environment that the tube-worm would see, and whether those bacteria in that biofilm would be ones that would be good inducers. Certainly, pseudo-alteromonads are probably 5 to 10% of the population. And we'd like to eventually go in to see whether MACs are naturally produced within those types of environments, although we haven't done that yet. But as I said, there are lots of other bacteria that induce metamorphosis as well. And those are ones, for example, the Rosiobacters, which are alpha proteobacteria. Those make up a pretty significant portion of the population, perhaps 40 or 50% in some cases. And those are really strong inducers of metamorphosis too. But it turns out that we've sequenced a couple of those genomes and found that even though they induce metamorphosis, they don't have any genes that look like MACs at all. And so it's probable that they have a very different mechanism of stimulating metamorphosis, and we just don't know about it yet. So that's another intriguing direction to take this work, is looking for the probably many, many different ways that bacteria are able to stimulate metamorphosis.
Yeah, thank you very much.
With that, I will thank you again, Nick, for teaching us so much about the scales of life and how they're all interacting with each other, from phage tails to animal development. Wonderful talk. Go have a good lunch. I'm assuming it's getting close to that time in your neck of the woods. And thanks everybody for coming. Cheers.
Thanks. Bye-bye.
Thank you.